The packaging of DNA is very complex. By unfolding and unrolling the chromosomes, the typical structure of the DNA molecule is revealed. A DNA consists of a double strand in the form of a double helix. Each strand is a string of 4 different subunits, the so-called nucleotides. Usually, these are named with their initials, A, T, G, and C. The DNA double strands are held together by chemical forces acting between the nucleotides, whereby at any time only A and T, as well as G and C, can build a bond. Both strands are complementary to each other, one being the negative print of the other. In terms of chemistry, the nucleotides consist of a sugar molecule, phosphate, and the nitrogenous bases. In terms of biology, the sequence of the nucleotides in the DNA determines the appearance and the function of an organism. A DNA double strand is composed of hundreds of millions of such nucleotides. Inside the nucleus, the DNA, packed into chromosomes, takes up the room of only a few thousandths of a millimeter. If all the DNA double strands of a human cell were unfolded and strung together, they would reach a length of 2 meters. Up to today, the entire DNA of only a few organisms has been deciphered, such as for man, mouse, cat, and dog, or the rice plant and grapevine. It has been revealed that the closer organisms are related, the more alike is their DNA. 96% of the DNA of man and chimpanzee is identical, which means that we only differ with regard to 4%. Thus, to identify organisms in the laboratory through their genetic information, those DNA segments have to be analyzed that are characteristic and unambiguous for a particular species or individual. If there are parts of arnica present in hay, its DNA can be found in the hay sample. Analysis in the molecular biology laboratory shall show if this is indeed the case. First, plant tissue is crushed in a bead mill. This way, cell walls and membranes are broken up. Then, the DNA is released into a solution. In order to get pure DNA molecules, a special procedure must be followed. This includes the addition of different buffers and several centrifugal steps. However, the DNA molecules alone do not allow any statements about the botanical composition of the hay sample to be made. But molecule biologists have a technique at hand that is used to amplify clearly defined DNA segments. This method has been copied from nature itself. A new double strand arises, but only a fragment. The copying procedure is repeated 30 to 40 times. until millions of fragments are generated. These are the so-called PCR products. Which apple variety is this? A Fuji? A Brabant? Today, most apple varieties tend to have a similar color pattern. Who can reliably distinguish these varieties? Nonetheless, variety denomination is of great economic relevance. It's all about patents and rights, labels and variety protection. Through genetic fingerprinting, a particular variety can be determined unambiguously. Thanks to molecular biological methods, apple breeding reaches a new level. Identification and paternity determination can already be performed on young plants, and not only when the trees begin to crop. With the aid of microsatellite analysis, researchers of the Molecular Biology Laboratory of the Research Center Limburg are able to determine apples that could not be assigned to a specific variety based on the study of fruit characters. Moreover, it is possible to reconstruct the family tree and investigate relationships. Such knowledge may be valuable. Due to their broad gene pool, old varieties become increasingly interesting for breeding purposes.